life.
Okay. What does it code specifically?
Genes.
Proteins.
Yeah.
Proteins. Okay, so DNA codes for a strand of DNA has three. Okay. Every group of 3 nucleotides on a DNA strand is called a codon, and these codons will be transcribed into RNA and then translated into a protein. So every 3 nucleotide will account for, will code for a single amino acid. And so there are 20 amino acids, and since there are 3, you have to have a series of 3 nucleotides. You would have 64 combinations of nucleotides to create amino acids, which means that there would be multiple codons for a single amino acid. And then these amino acids code for proteins, which determine your, like how you look and what your cell does. So this is important to know in the case of mutations, because if something is mutated in here, it could code for a totally different amino acid, which could mess up the protein sequence, or it could code for the same amino acid and not change the protein sequence. So there's, it could be a silent mutation that's in the DNA, or a drastic change in the protein. So it's protein. Mutations. So there are 4 different types of mutations. Okay, wait. So what is a mutation? A mutation is a change in the DNA, and because DNA affects how an organism looks, behaves, and its physiology, a change in the DNA can affect all parts of an organism's life. So there are 4 different types, or different types of mutations are based on how the DNA is changed. These types are substitution, insertion, deletion, and frame shift. And in substitution, a mutation happens when an incorrect base is placed in, or used in place of the correct base. So in the example of guanine GCA, when that gets replicated into the into RNA, it would be CGU. But when it gets replicated, it could change this A into a T and code for a totally different amino acid. And if an incorrect amino acid is placed into a protein, it could change the way the blood runs, like in sickle cell anemia. The substitution could potentially change a codon into a different amino acid, like I said, creating sickle cell anemia, or the codon could be changed to code for the same amino acid, where so an amino acid for alanine is an amino acid, has 5 or 6 different codons that will code for that amino acid. So this is a silent mutation, and nothing has changed. And then instead of coding for an amino acid, it could change where a stop codon is, producing an incomplete protein, and so there's no function for that protein. Insertion is where an extra base pair is placed into the sequence. So instead of having a multiple of 3 in the series, you would have maybe an odd number like 13 or 14, and this too can change the sequence and how RNA reads it. And then the deletion is where, like it sounds, a codon or nucleotides are taken out from the sequence, and these two, both insertions and deletions, will cause a frame shift, and a frame shift shifts all of the way that the DNA is read into groups of 3. So if something is deleted from so these are all codons, and if one of these nucleotides is taken out, all the codons will be shifted a little bit, and you'll have an incomplete codon in the final area. And so all these shifts will it will mess up all the synthesis for proteins. And then there are 2 different types, or mutations can happen in 2 different types of cells: the reproductive cells, or the germ cells, and these will be passed on to the next generation. And somatic cells, or like cells that aren't responsible for reproducing, like your skin, and these will only affect the individuals. So cancer tumors are a good example of these non-reproductive mutations, whereas hemophilia is a mutation in a germ cell. So where and what? So somatic versus germline. So somatic is and germline is so there are many reasons, or there are reasons for why mutations happen. There are natural causes and external causes. Natural causes can happen during cell division, and the copies aren't exactly perfect. And as we know, DNA polymerase has proofreading abilities, but sometimes they don't catch all of the mistakes that happen. And so we get natural causes to mutations, which may potentially be a harmful mutation, or it could be a silent mutation that doesn't change what the DNA is coding for. And external influences or external causes are like exposure to radiation or chemicals that result in the cell doing an imperfect job and resulting in the DNA that's mutated. And with that being said, DNA mutations are random in most cases. And the environment around an organism can affect and increase the rate of mutation, but it's not one of the reasons why a mutation occurs. So when an organism is resistant to a chemical, it would be because the organism has already had that mutation to be resistant, and it's not the mutation isn't made to benefit the organism. And so most mutations are caught at the time through like the proofreading and the endonuclease activity, but not all of them can. And so there are flaws and mistakes happen. So the effects of mutation. So some mutations can help with evolution, such as resistance. But in a germ cell, a mutation that can be passed off to the offspring, and there are no changes. But when there are changes, they can be of little effect, or they can change the phenotype so much that death can occur. So one of the questions is there a germline mutation that you can think of before we go into beneficial mutation?